Hello everyone, I am Salman Ibn Yunus. In this presentation, I will review a paper that was published in Oxford Academic Journal. The name of the paper is Pattern Recognition in Bioinformatics. So, first of all, we need to know what is pattern recognition. Pattern recognition is concerned with the development of systems that learn to solve a problem using a set of example instances, where each instances are represented by a number of features. For example, this problem could include clustering, the grouping of similar instances, classification, dimensional trees. Dive into the presentation. So let's start with the introduction of the paper. So as we know, there is a huge increase in the volume of sequencing data such as DNA or RNA or genomics data produced lately, as the cost of next generation sequencing for genomics and transcriptomics has dropped significantly over the last few years. As a result, there is a rapid growth of bioinformatics over the past decade, and there is a requirement of computational tools to manipulate and handle to make sense of sequencing data to produce or infer insights from the sequencing data. So, pattern recognition has been highly successful in helping to explore and exploit high throughput measurement data such as sequencing or genomics data. So, we can use pattern recognition to gain insight from those genomics data or DNA or sequencing data. So, pattern recognition means looking for specified patterns in sequences and looking for novel patterns. Therefore, since the analysis of high throughput data are becoming common to modern biology, for this reason, bioinformatics and computer scientists must learn about pattern recognition or machine learning and apply those techniques in for predicting bioinformatics or other related problems. In recent time, lots of studies are being conducted using various classification and clustering algorithms to investigate bioinformatics problems like transcription factor binding, histone modification, or chromatin accessibility, etc. Let's look deeply and to know what is pattern recognition and how it can be applied. Pattern recognition follows a series of computational steps to achieve the goal. So the starting point, as we know, is usually the collection of dataset or training objects, which is assumed to be representative of the problem, and thus for new objects to which the system will be applied later. The first stage consists of translating raw measurements into data, which can be used for further processing. Some pre-processing steps can be handled by measurement device or accompanying software. Further pre-processing techniques depends on the problem and on available prior knowledge. In case of sequencing data, quality inspection is also important to avoid problems in subsequent analysis, and also outliers should be detected and removed. After pre-processing, measurements are adequately represented, usually as features or feature vectors. Then a subset of informative features are selected, which are used to train a classifier. How the data are represented in pattern recognition problems. Let's take a look. So, having a good representation is the most important steps a satisfactory towards a satisfactory towards satisfactory solving a pattern recognition problem. We need to we need to take a look so that we need to be careful so that too much information is not lost in this step. Good performance will be impossible to obtain if too much information is lost in this step. Most pattern recognition algorithms assume that an object is represented by a feature vector x of real valued numbers. Recently, a new development has come which is driven to a large extent by application in bioinformatics. This is that of representing object pairs xy by kernels xy. So, what are kernels? Kernels are functions that, under certain constraints, allow relatively simple algorithms to operate on complex representation objects in high dimensional feature spaces and doesn't need the need ex- to calculate those representations explicitly. Moreover, the development of support vector classifier, which derives much of its powers in application from using kernels, was used in a large number of proposals for kernel and kernel-based methods for bioinformatics. A classifier. Let us take a look at some of the most common pattern recognition algorithm. There is clustering, dimensionality reduction, classification. So, what is clustering? Clustering is basically natural grouping of data into, for example, instances into groups. Such groups may lead to new insights. So, we explore a large data set and find whether there is a distinct subgroup of similar objects. For example, genes whose expressions over a range of microarray experiments are similar may be functionally related, and the Gilbey association principle can be used to infer the function of uncharacterized genes. Likewise, proteins are clustered based on sequence similarity to learn about orthology, organisms based on genomic similarity to find phylogenetic trees, etc. Dimensionality reduction. Dimensionality reduction can be performed in both supervised and unsupervised learning. So here the objective is to decrease the number of features while retaining as much as information as possible about the classification or regression problem which solve later. Thirdly, there is classification. This is the most widely studied question in pattern recognition, which is to how to design classifiers and how to assign algorithm labels to previously unseen objects given a training state of objects x with accompanying labels y. So these algorithms can be used in patterns. So an important step in pattern recognition is called performance evaluation. So this is very important because to evaluate how well a model will perform on new objects in case of supervised pattern recognition problems. It is important to take prior probabilities into account in classified design and evaluation when possible. So in bioinformatics application, prediction error is not always the most informative criterion, especially in problems where the main aim is to distinguish one target class. So performance estimate based on the objects used to train a system will be biased. This is because the system has seen those objects before, so they are no longer representative of new objects. So evaluation thus requires keeping apart objects or examples that can serve as a test set, not previously used in any way to train the system, set parameters, or make choices. So performance evaluation is not only the last step in pattern recognition pipeline, but also used for intermediate steps as feature selections. Some of the recent developments in pattern recognition are explained in this slide. So these are some new models and algorithms. So the first one is bag of instances representation of objects. So in multiple instance learning, the problem is considered where such a bag is level positive if at least one instance is level positive. This is already found application in drug discovery, relating possible structure of molecules to activity and predicting protein binding sites. Secondly, there is semi-supervised learning. In semi-supervised learning, It can be applied in cases where there is a large number of unlevel objects are available besides a small set of level objects. This has already been exploited in prediction of gene function, in expression-based clustering, in the prediction of transcription factor binding sites. There is another new model or algorithm called structured learning for predicting arbitrarily shaped output rather than a single level. Methods include hidden Markov models and more recently structured support vector machine and conditional random fields. They have been applied for predicting gene structure, protein secondary structure, drug activity, etc. There is another new development called active learning, which is a classifier is used to decide which unlabeled object should be labeled next to best improve the classifier. Application already explored include diagnosis, gene expression sampling, drug discovery, and predicting protein interaction and transmembrane helices. Active learning is often used when classifier predictions are ranked and the most confident ones are verified experimentally first. Some of the recent developments in next we will talk about some of the major challenges we face in case of developments in pattern recognition. So there is new challenges in biology also promise to place new demands on model and algorithms. So nowadays it is becoming challenging to tie together various heterogeneous data sources in a single application. Pattern recognition algorithm will have to be more robust to missing data. They will have to be better able to deal with various types of data and scalable to many more objects. Given limited storage and bandwidth, algorithms may have to be able to work on compressed or summarized data. Secondly, as tools for measuring and particularly manipulating the cell become more widely available, pattern recognition should help close the system's biology loop by supporting researchers in setting up experiments. Given a limited experimental budget, which interventions together with which measurements are likely to increase our knowledge most. So in this case, active learning may prove very useful. Lastly, it is important to help user to interpret why a pattern recognition algorithm predict what it does. This is often called as a black box algorithm, which allows for and it can move from black box to gray box model, which allows for a gain in biological knowledge. This requires a shift in emphasis from the performance of a trained predictor to its makeup, stability, and uniqueness. Lastly, to conclude, we can say in this review paper, the authors have covered the core elements of a pattern recognition course, which includes data representation, the problem of clustering, dimensionality reduction, and classification. Also, performance evaluation, model selection is discussed. They focused on the major issues and potential pitfall in applications and in interpretation of the results. That's all from today. That's all from this video. Thank you for